So this is something which I had been working on in this field for the past 1 year as part of my MSc project. And based on that, whatever few things I've learned in terms of systems biology, I'll try to share a glimpse of that. And hopefully more people come into systems biology, especially chemists, because many people don't actually know what systems biology actually stands for. Uh, okay, so let's get started. We'll be following this particular course through 5 days, and I'll try to give you a flavor of systems biology as far as possible. Uh, some basic introduction. My name is Alok Apam Swatiputra. I completed my BSc from this very room itself, room number 35B, and then I moved on to IIT Bombay to pursue MSc in chemistry. And the picture which you see, the background is from Sami Hill inside IIT Bombay. So we have a mountain inside our campus, and it's almost a ritual that the first thing you do after going to Bombay is climb up that hill and take this particular photo. Now, why I put this particular photo is because one, I graduated from the institute pretty recently, and also you can see the different sort of facets which nature can provide us. As you can see from this one picture itself, there is an entire ecosystem. You can see there is a water body, there's a hill, there's forest area, there are buildings. So it encompasses all the possible sort of things which you can see in natural environments or nature in and around you. And nature follows very strict grammatical rules. And systems biology, the basic motto of systems biology is trying to decipher the grammar of how nature operates. How does nature utilize the grammar of mathematics to build upon the literature of science? Now, some nostalgic here, because since I showed IIT Bombay, I should show this picture as well. So this is taken almost 2 years back in April of 2022. Not all the professors are there, but most of them are there. As usual, IC sir in photos look somewhat like this in almost every photo. And you can see the others as well. And these are my batchmates. So being on the other side of the benches is a bit awkward. I'm usually more used to being where you are. But okay. My only achievement is I managed to escape the college 2 years before you could. I guess that's there. And that's why I can give you so much gyan. Hopefully. Okay, so let's get started. Some basic skill and modalities of the course. Uh, since today was the first class, we got a bit late, so that's okay. From tomorrow, we'll try to stick to schedule. So from 2 to roughly like 1 hour, 1 hour, 15 minutes, I'll try to give you the instructions. Then we'll have a short break, and from 3:30 roughly to around half an hour, 45 minutes onwards, we'll do some problem solving and tutorials. Okay. Modalities classes, as usual, will be held offline and over Zoom simultaneously. And since the meeting is being recorded, we'll upload it to the YouTube channel as well. So you can refer back if you want, if you have missed any part. Yes. Now, full disclosure at first. First of all, you can contact me through my Gmail ID anytime you want. I'm free all the time, at least for now. And the most important spoiler alert, you might not find any chemistry in this course. So there will be no benzene molecules. You won't see any octa-coordinated, hexa-coordinated complex. No sort of what you can say quantum chemistry here. But stay tuned with an open mind. Even if it is not objectively chemistry, you might try to appreciate the underlying chemistry which we try to discover through this course. So hopefully you won't be disappointed. So what is the basic philosophy of the course which we'll be following? So there will be some writing involved. So you can see at least in offline, most of the people have some writing material with them. And for the guys who are attending online, please try to keep some pen and paper with you if you deem it necessary to jot down notes if you want. Interact freely without hesitation. I am not a professor. As I said, my only achievement is I passed out 2 years before you. So let's learn together because I have also come into this field and this topic just 1 year. So even I don't know a lot of things. So whatever I don't know, I'll try to learn from you. And whatever I have known, I'll try to give you a flavor of that. And please stop me anywhere in between. You don't have to wait whether I'm speaking, I've completed a topic or not. And there are no stupid questions because most of the people don't have an idea what the topic is, so there cannot be any stupid questions. Every question is a valid question regarding this. So this is the basic philosophy of the course and how it will be followed. And what can you learn from this? So first of all, you'll get to see how quantitatively you can start to understand biology. So you'll develop a quantitative mind, not qualitative, but how much, what is the time scale in which biology operates, what are the length scales in which biology operates. You can also start to appreciate the design architecture which is present in nature. Usually, when we see things from a bird's eye view, a broad perspective, it is not immediately evident that whatever we are seeing, how exactly the inner workings have been worked out across evolutionary times. There are very strict rules which have been followed, giving rise to the macro phenomena which we are observing. So I guess you'll start to appreciate the intricate design architecture. Also, you'll get to see an idea of how interdisciplinary research works out because systems biology involves research from every field possible. There are physicists, there are chemists, there are biologists, there are doctors, and every possible field of science is required to do systems biology. And hopefully, you guys will develop an interest in systems biology. And in the worst case scenario, if none of this happens, you will hate me for wasting 5 days of your life. But that's okay. So let's get started. Since we are talking about systems biology, what is a system in biology? Now, if we look at this for chemists, when we talk of a system, you usually refer to a thermodynamic system where there is the system, boundary, surroundings, and the entire universe. So there are different types of systems. You can have open systems where both mass and heat transfer happens. You can have a closed system which does not allow mass transfer but allows heat exchange. And you can have a completely isolated system where nothing is allowed. No mass transfer and no heat transfer. So I would like to pose a small question here to whoever is attending and here offline. Can you give me an example of an isolated system? Yes. This is a classic question, which is usually tricky, but I guess the Vedians know it very well because this is taught very well. The only example of an isolated system is the universe. Always. There is no other isolated system. So for chemists, the thermodynamic system is usually what you are accustomed to. Now for a biologist, what is a system? Now for biologists, system looks somewhat like this. And the picture might be very complicated, but if I explain to you in terms of language, you have evidently or subconsciously or consciously used the word system at least if you have studied biology anytime in your life. Have we not used the term circulatory system, respiratory system, immune system, skeletal system, muscular system, vascular system? So every unit of biology can be defined to be a system in a sense. And what is the basic sort of underlying fundamental features for a system in case of biology? Usually, biology works in sort of a signal and output sort of thing. So, for example, this is a picture of an immune system in a particular bacteria. If you provide stimulus, which is either the bacteria is interacting with its environment, some antibiotic has come into the environment, suddenly there's a temperature jump. So all these are some sort of signals to the bacteria, right? It will start sensing these signals from in and around the environment. And consequently, the system will start responding to the particular stimulus. And that is the output. So essentially, so every system in biology is sort of a signal processing unit. Yeah, because the system is in its place, and what happens is, uh, so you put a signal as stimuli as a signal, and you will get the corresponding output. So every system in biology is sort of processing signal as we go through it. It starts sensing the signal, then it processes it through its in the internal machinery, and once the signal has been processed, it will give the corresponding output. Now, as you can see here, this sort of a system looks very complicated. Nothing is here. There are like some random arrows here. Some are pointed, some are sort of dashed arrows, some are hammerhead arrows. So the question is, how do we study such complex systems? Because with a thermodynamic system, if I give you the pressure, volume, and temperature number of moles, you can describe most of the macroscopic problems. But if I tell you the concentration of one of the proteins which is present in this particular unit, it will be very difficult for you to tell me what is the corresponding output or what are the expression levels of the other biomolecules in there. So how do we study such complex systems? So there are two approaches which are possible. One is called a top-down approach and one is called a bottom-up approach. Now let me give you an example. Suppose you are preparing for your end semester exams. One possible mode of preparation is if I give you access to the library, you have access to the entire information system of the college. So all books are accessible to you. And also you have access to the previous year question, which is your output. Now in the top-down approach, what you do is, since you have access to the library and you also have the previous year questions, you can find out the important books, right? Yeah. So you can find out the important books, but you have to search through the entire catalog of your library to find them. So this is the top-down approach where you have access to a large amount of data and you know what your sort of desired output is. And consequently, from the large data, you start breaking it down into small pieces and analyzing, and ultimately go to your output. Yeah, so that is the top-down approach where you have large data set access and you know more or less what your output should be, and you start breaking down and analyzing the large data set to get your ultimate output. The second mode of preparation is what you can do. If I tell you what the important books are, and then if I tell you what the important books are, you can go through the books. And as you go through the books and start solving problems, you can generate the entire important questions even if you don't have access to the library, because you don't need the entire data set to work with. I have given you already what are the important sort of years which are possible. It's more or less physics. But there are not much fine print here, so it will be fine. There is not much fine print of small font, so it will be. Yeah, so sorry for the interrupt. So just to revisit, yeah, for the top-down approach, you have access to an entire large data set, and you know what your output of questions are across the years, and you start to break it down and find out the important books. This is the top-down. And in the bottom-up approach, if I tell you what the important books are, and if you go through them enough number of times and start solving problems, you can start to generate the important questions. And then you don't even need the previous year questions access. You can actually generate the important questions and the previous year questions because you have followed the standard books only and not gone through the entire dataset search. So this is the bottom-up approach. And this is how the modeling in systems biology also works. So it's a breaking down versus building up. So in terms of the top-down approach, if you have access to organism level information or like high throughput sequencing data, you have the entire genome sequence of a sort of animal, you can start analyzing it through computational techniques and then cut down, focusing on the important things and ultimately reach to a level where you know exactly what protein or what gene or what RNA is important. And then you can figure out, okay, that this is the gene I should target in terms of therapeutic purposes. And in a bottom-up approach, you do literature survey, you do small-scale experiments, you figure out what the important genes, what the important proteins, what the important RNA sort of things are, and then you start to look through literature and through experiments in a certain way and start to build up a model that will be robust enough to generate organism level behavior when you simulate through the entire model. So this is how modeling works. So in top down, you have a lot of data sets, you break it down into the most important parts. And in bottom up, you take the most important parts and build it up into an organism or cellular level or whatever high level behavior you. So in our particular discussions, we'll be focusing mostly on the bottom up approach. The bottom up approach involves the beautiful synergy of experiment and theory, and you have to do this hand in hand. So the main aspect of bottom up approach is you decide to do experiments depending upon what do you want to study instead of a high throughput data, which is in case of a top down approach. Here you have to be very specific that what question do you want to ask, what is it that you want to look at, what biological interaction are you trying to look at. Do you want to look at genes, then you have to do genetic experiments. Do you want to look at biomolecular networks, then you have to work with proteins and mRNAs and other involved enzymes and all the sort of lipids and all this sort of stuff. If you want to go one level up higher and you want to look at cellular levels, that how do cells interact or cells communicate with each other, then you have to do cellular level experiments where you have to do cell culture experiments and sort of things where you have multiple cells and how they are interacting with each other. And ultimately, you can even go up to the organ level network. So depending upon what sort of level of network you want to work at, you have to design your experiment accordingly. Now, how do such experiments look like? For example, if you want to look at, suppose, say, a molecular level, what you do is you do a sort of protein or a mRNA expression. You can see that this is some equation and alpha tubulin protein. This is a Western blot which helps you quantify the amount of protein which is present in your system or cells rather, collection of cells. You can also do cellular level experiments, and you can see the ones which are colored red here. These are actually cells, and they are colored red because they have been sort of incubated with something which acts as a marker of cell cycle. So in which phase of the cell cycle they are, depending upon that, they will be colored accordingly. So experiment will give you the measure of certain physiological outcomes, right? You can get mRNA levels, you can get the dynamics of temporal dynamics of protein, you can start understanding the cellular dynamics. What is the cell division time? What is the cell motility? How fast are the cells moving? How fast are they changing their phenotypes? They're undergoing differentiation. So this is one part of the experiment. Now, once you have certain experimental data in hand and combine that with literature, you can start building up small models where you take the most important frameworks and you start looking at how do they interact with each other. For example, you take three sort of proteins or microRNA. So NIC is a transcription factor, which is some sort of a protein. MicroRNA is RNA. E2F1 is also a transcription factor. So in a sense, it's a protein. So you identify that, okay, I want to look at these three particular things and how do they interact with each other. So either from literature or from experiment, you'll find out most of the time. You find out that MIC actually increases the amount of microRNA. So you put a increase. Now E2F1 also increases microRNA. So you put that interaction again. But microRNA inhibits it. So whatever is hammerhead in this particular picture is an inhibition or a degradation sort of process. Whatever is a straight normal error, the activation of promotion. So once you build a small model like that, essentially what these are species interacting with each other, right? These are chemical reactions. So we have all studied chemical kinetics, so we can start writing equations like this. These are not as simple as the ones you will encounter normally in chemical kinetics, but I assure you the essential chemistry underlying them is actually this is also pretty simple. I think you can appreciate that whatever is raised to the power n here, most of you must have seen such a term in case of a hemoglobin binding dynamics, where you have a Hill coefficient. So hemoglobin binding dynamics, when hemoglobin binds with oxygen, it follows a saturation kind of dynamics. So initially there is very low, and once cooperativity rises in, you have a Hill coefficient, which is slightly greater than 2, I guess, for hemoglobin. So these terms are usually known, and how you combine these terms is important. So once you combine these terms, then start the back and forth affair between your experiment and theory. So in terms of this equation, you can see there are many rate constants, there are these coefficients. Either if you found them from literature, that's well and good. Someone has already done the experiment for you. If you don't know what the rate constants are, you can do the experiment, find out the rate constant, put it back into your model, and see if the updated model can regenerate your experimental phenomenon. And once you do this process enough number of times, at every iteration of this process, you are enhancing the robustness and flexibility of your model. That how good is your model at replicating the experimental features. So this constant back and forth affair between your kinetic modeling and your experimental sort of outcomes will help each other in better generating your model. Essentially, you are trying to regenerate experimental phenomena by doing more experiments. That is the main aim of your model. One word of caution which must be mentioned here, all models are wrong. Your model is only good for the dataset you have generated. There is no model which can encapsulate every biological behavior in the world, for every chemical behavior for that. So always be careful when modeling anything you do. Your model is good for the data you have chosen. Unless and until there is independent verification from number of people, you cannot provide sufficient sort of backing to your claim that this will regenerate every phenomenon, or I have the ultimate model which I have generated. No model is ultimate. Every model will be proven wrong in some time. But you can generate a good enough which can represent most of the phenomena. And once you generate a robust enough model, what you can do is you can also start predicting certain experimental outcomes which might be possible. For example, you have a certain protein which is not possible to be extracted using the current technologies which are. But since you have developed a model for it based on the interactions, you can study theoretically using a computer what their expression profiles might be, what is the level that it might reach in cells. 10 years down the line, 15 years down the line, when the protein is actually extracted and the technology develops to that end, you will see in most of the cases, as it has happened in the history of systems biology, it has agreed well with the theoretical predictions, which were rejected by the experimental biologists maybe 10 years, 15 years back, but ultimately that turned out to be true. So it's a very powerful tool that if you don't know something experimentally, you can predict it, and someone will do the experiment for you down the line and will verify your theoretical prediction. Okay. So since we talked about sort of incorporating a lot of maths in terms of chemical equation and differential equation, let us look at some of the evolving roles of how mathematics and computers started to come into biology. The very first case was in case of theoretical biology, where there were no predictions and those sort of stuff. What essentially happened was they started to look that can I prove my experimental fact based on a theoretical method. What they're essentially trying to do is you have an experimental fact. You're saying, okay, the protein in level in a cell oscillates after every 20 hours. So it goes up and comes down after 20 hours. Again, it will go up and come down after 20 hours. So can you generate that from a theoretical model where you only have the concentrations and chemical kinetics and you simulate it on a computer, and if you manage to show that, okay, even theoretically, if these interactions are present, then the protein will oscillate after 20 hours. So that is how the field of theoretical biology actually started. But experimentalists did not put much faith into that because nature doesn't believe in your theory. Nature will do what it has been hardwired to do. It is your job to prove whether your theory is correct or not. It is not nature's job. You have to provide a good enough model, and that's why it was very difficult to convince the experimentalist initially. Because when you think of biology, you always think of experiments. You think of animals, you think of plants. Nobody used to think of maths in biology. But slowly that has changed because of what I mentioned, that the predictions which were made using mathematics eventually turned out to be true in terms of experiments as well, 10 or 15 years down the line. And that's when the experimentalists also agreed that theory can become a powerful tool in terms of experiment, in terms of designing the experiments, in terms of predicting, or in terms of at least having a know-how that what should I expect from my experiment instead of blindly going into doing an experiment. And after that, it slowly progressed as more and more computational power came in, more and more mathematicians, engineers, physicists became involved in the problem. And that's how, from theoretical biology, we moved on to computational biology, bioinformatics, which I guess most of us have at least heard bioinformatics and computational biology. And from that, systems biology work, which takes a look at biological interactions across many different components, and so there is a lot of difference in time scale, length scale, every sort of scales which are possible in biology. Systems biology tries to put a macroscopic bird's eye view of what is the overall picture which we are trying to look at, and how are the individual components in that overall picture interacting with each other. And the ultimate uh, destination which has been reached till now is called molecular systems biology, which looks at the interactions of individual macromolecules at the molecular level in terms of how they are getting phosphorylated, for example, in terms of physical significance, how one enzyme is catalyzing another process, how one enzyme is being inhibited by another sort of inhibitor when you add a drug in terms of drug designing, and then starting to look at the interaction. So bioinformatics, along with computational biology, structural biology, and systems biology modeling, ultimately gives rise to the most minute interactions you can look at, and that is called molecular systems biology. And in the age of AI and chat GPT, I guess most of us are at least somewhat aware of computational biology because we have all looked at like AlphaFold protein folding experiments. The latest one is AlphaFold 3. You can also do high throughput sequencing experiments where you can study the RNA expression, DNA expression of 10,000 cells and 20,000 genes at one shot in one experiment, and then use computational data analysis to generate protein. So this is how mathematics and computation slowly crept into biology, and biologists also started to gain more confidence that we should start using mathematics and computers instead of blindly relying on experiments only. Because doing experiments in biology is much more costly than doing a simulation in biology. Okay. Now, since you have mentioned the bottom-up approach we'll be discussing, there are two possible ways of doing a bottom-up approach. It is called, or rather, modeling a particular system or a particular interaction. One is called a phenomenological modeling, and the other is called a data-driven modeling. How do these two work? A phenomenological modeling scenario, you have some idea or a pretty good idea in terms of literature and what has been published. You have a good idea of what your network of reactions look like. So, what are the species involved? What degrades what activates what phosphorylates what inhibits what. You have more or less a good idea of who are involved and how are they involved. Now, since you have the reaction network in hand, you can write down differential equations in terms of chemical kinetics, as we have already seen. And once you solve these equations, either analytically or numerically, whichever way you want, usually numerically using a computer, you can start to see the dynamics of signaling, that how the concentrations of these species affect each other. What is the temporal dynamics? How does X affect Y? How does Y affect X? If X is present, how does the timing of Y expression change? So you can start to look at the dynamics of systems, and you can also start to do parameter sensitivity analysis, which we'll look into later in this. So this is the phenomenological model where you know that these are the phenomena which are present. You start writing down equations for each of these phenomena, and then you regenerate the dynamics of the system. In the data-driven model, what you do is you collect a large amount of data, and then what you do is you do computational analysis. So you classify, okay, so you have, suppose I say, for example, uh, the gene expression levels are 10,000 cells. Now, some of them are cancerous, some of them are non-cancerous. So if you do computational analysis, based on the gene expression profile, you can segregate that these are my cancerous cells, these are my non-cancerous cells, these are some sort of intermediate cells. So these are all sequencing, mostly high throughput data, which is basically done on a computer. It is not possible to do it by. So this is the big data age of biology, which is mostly used by the top-down approach. Because you see, you have access to a large amount of data, which I have mentioned, and then from that large amount of data, you identify the key signaling pathways and you start to predict the phenotype. So you're breaking down large data into smaller chunks, which are the most important ones. That is the top down, and we'll discuss the phenomenological model because that is more important in terms of the bottom up approach, where you know what species are important and you start to build up a network which will give rise to physiological phenomena. Now let us do some road trip. How do we model a biomolecular or a biological process? So I think most of us have gone through this treatment at least somewhat, depending on at least the third years have definitely done this. Not sure about the first one. So let's revisit the data. Michael is here. So let us go through the revision once again. Even if you haven't done, we can just revisit it once again. So you have a enzyme which is reacting with a substrate. When it binds, it forms the enzyme substrate complex. Now the enzyme substrate complex can either give rise to the enzyme and the product which you want, or the enzyme substrate complex can dissociate back as well. And you have the corresponding rate constants for each of these particular processes. And what you can do is, so if I write the rate of the equation, which I denote with V, if you term the rate of the equation as, suppose say, K2 into product concentration, right? Because that's how fast your product is being formed. Oh, sorry, not K2 into product, but K2 into enzyme substrate. Sorry, enzyme substrate. Because this is how fast your product is getting. Now what you do is, you assume that both K1 and K2 are much faster than K minus. You take a very broad assumption, which is usually a good enough assumption in terms of biology. And what this enables you to do is apply steady state approximation. because the enzyme substrate complex is getting formed extremely fast and also giving rise to the product extremely fast. So it is a very short-lived intermediate. It will rise up to a certain level and then the things will balance out from both sides and it will maintain a constant level. So once you apply steady state approximation, what you can do is you can say that the change in concentration of enzyme substrate complex at steady state is zero because it has attained the constant level. Now we have to write the differential equation for the enzyme substrate complex. So what are the processes which are involved in the enzyme substrate complex interaction? One is it is formed at this rate from the enzyme and substrate. There's a negative K minus 1 ES because it dissociates back into this process. And also there's a K2 into and we set this equal to 0. Now in terms of biology, since the enzymes work very fast, it is difficult to measure the free enzyme concentration at any particular time. So we'll do a small algebraic trick here. Since the total amount of enzyme you added at the start of your reaction is finite, what you can write is the total concentration of your enzyme at any time will be the free enzyme plus the enzyme which is bound to your substrate complex. Right? We can write this because you haven't added anything externally. Either whatever is free plus whatever has been bound is what you added. So we can replace this E here. Right? So we can write this. Now if we take certain terms common, you'll see we can write K1 total substrate. And here, see the concentration of enzyme substrate, we can take it out common because it is present across all the other terms. So if we take it like this, what is left? There will be a K1S and now what we do is we move all the terms to the other side and keep it here. So this becomes K1 S divided by S. So we have found out what is the expression of the enzyme substrate complex at any time in terms of the total enzyme which we added at first and the substrate concentration at any particular time. And this is easy to measure because you cannot measure the enzyme substrate complex because it is so short-lived, but you can measure the total enzyme concentration, which you know what you have started with, and you can measure the substrate concentration left over at any time. Now, since we have found ES concentration, we can write the final rate as K2 into ES, so it becomes K1, K2, S, I. I'm just hearing a few terms here. Yes. Right? And if I divide the entire numerator and denominator by K1, what you will be left with is K2 total enzyme substrate by K minus 1 plus K2 by Q1 plus, right? And this is what all of us have studied, that the rate is max this is a Michaelis-Menten equation, where Vmax is K2, which is the enzyme substrate to the product formation rate constant, into your total enzyme concentration, and your Kn is K minus 1 plus K2, right? Rate at any time. So if you do this particular experiment where you take in a total amount of enzyme and you add substrate and you follow across different substrate sort of concentrations, what is the rate of the reaction? You can do this particular experiment. You'll see that this is the sort of curve which you observe, because from this equation, which is Vmax S by Kn plus S, this is sort of the profile which you'll observe. And most of you must be knowing that this Km is the concentration at which the rate is actually half of your V. Because you can try it yourself as well. If you put sort of what Km as Vmax by 2, or what you can do is you can put V equal to Vmax by 2. So put V equal to Vmax by 2. So Vmax will cancel out. So you will get 2Km, or sorry, put V equal to Vmax by 2. You will get Km plus S equal to 2S. Okay. So from that, you can say that K equal to S at P equal to V by 2. So this is how mostly the Michaelis-Menten constant, which is Km, is figured out. You do a profile of the rate of the reaction across different substrate concentrations. And then whatever substrate concentration, your rate is half of your maximum rate. That is your Michaelis-Menten constant. And physically, what does the Michaelis-Menten constant of an enzyme signify? If you look at the expression, it is K minus 1 plus K2 by K1. So in the numerator, whatever terms are there are either the dissociation of the enzyme substrate complex or its formation into the enzyme, free enzyme and the product. And in the denominator, there is the sort of binding of the enzyme and substrate to give rise to enzyme substrate complex. So in the numerator, you have terms which are killing off enzyme substrate complex. And in the denominator, you have terms which are forming the enzyme substrate complex. So from here, what can you identify if I say that the Michaelis-Menten constant of enzyme B is higher than that of enzyme A for the same substrate? Okay, okay, might be see, in terms of catalytic activity, it will be difficult to mention because you have K minus 1 and K2. So catalytic efficiency will be K2, but it can happen that K2 is high, sufficiently high, but K minus 1 is much higher. Then you don't have product formation at all. Short, what can you tell us in terms of the binding efficiency of the enzyme? How strong is the enzyme bound to the substrate? So since all the dissociation, killing or degradation terms of enzyme substrate complex are in the numerator, the one which has a lower K will have lower degradation and dissociations. So that enzyme will bind more strongly to the substrate. So we can say higher the Km, lower is the affinity of the enzyme for the substrate. Make sense? So this is a good way you can measure the performance of enzymes on the same substrate. You can do this particular experiment across a series of enzymes, and then you compare their Km value and you say, okay, this enzyme has low binding affinity for this substrate. This enzyme binds to this substrate more strongly, and this particular. So you can sort of compare the efficiency, or rather the binding affinity, at least to a certain extent, of enzymes and substrates. Now think carefully of what you have done. This is a very powerful technique. Even when we study chemical kinetics, we don't realize that through this one mathematical equation, we have encapsulated the behavior of 10 to the power 23 number of molecules. So you have taken an Avogadro number of molecules, and without any idea of where they are, at what position in the biological system they are, you have managed to generate the at least the macroscopic, the thermodynamic behavior, or rather the kinetic behavior, I should say, not the thermodynamic, but at least the macroscopic kinetic behavior of Avogadro number of molecules by one mathematical equation. So that's the power of mathematics when applied correctly to chemistry and biology as well. This is the simplest possible biomolecular process which I have shown, which is the enzyme substrate binding. Now, armed with the knowledge of this, let us take a look at one step further, and things might get a little bit more complicated from here. So please stop here. What we'll do is we'll apply Michaelis-Menten kinetics to a bacteria growth problem. So you have a reaction vessel which has a fixed volume V, and you're flowing in nutrient material from one side and extracting it out from the other side. So you have a flow rate which is in terms of liter per second. So certain volume, suppose 10 liter per second, you are flowing in through the reactor. And there are certain bacteria which we call microbia in your reactor. Now the bacteria, once they find nutrients, they will start growing. They will start dying. But let us take the simplest possible process. So you will have some bacterial density at every time point, and I have defined time as capital T here, but we'll change it shortly as you will see. But let us, for this step at least, assume that capital T means time, not temperature. Okay, so it's time point. So at any time T, you have a certain microbe density or bacteria density, which is number per liter. Suppose 10 bacteria per liter or 1000 bacteria per liter. That's your microbial density. And you have your nutrient concentration because you're flowing in nutrients into the system. And since it's a chemical, you can write it in terms of moles per liter at any time. Say there is 2 molar nutrient concentration. Okay. So this is the overall system. You have a tank which is of a fixed volume V. You're flowing in nutrients through that, and bacteria is growing inside that tank. How do we write down the equations for such a system? It might seem a bit complicated, but I'll explain the terms as we go. Since we have used Michaelis-Menten kinetics in the earlier problem, let us apply it to this problem as well. So, look at this carefully. You can write the microbe density changing as a function of time. So, the first term here is a Michaelis-Menten term. So N here is the nutrient concentration. K is the nutrient concentration at half maximum. So this is simulating what the Km in terms of your enzyme was, but instead of the substrate concentration, here you have the nutrient concentration which is available for the bacteria. You have certain R, which is the maximum possible reproduction rate of a bacteria. Okay. So if you look at it carefully, the entire first term here, there is a n by k minus n. There is multiplication by R, which is the reproduction rate of bacteria, and there is n. So if you think carefully, more the nutrients, more will be the bacterial growth. More the reproduction rate, more will be the bacterial growth. And more the number of existing bacteria, more will be the bacterial growth because there are more number of bacteria to divide. So this entire term is positive and it's multiplicative, and we have kept the Michaelis-Menten term to take care of the nutrient flow. Also, since you're flowing through the system, some of the bacteria will flow out, it will get washed out of the system. So if you have the flow rate here, so if you take F by V and multiply it with the number, so suppose the number of bacteria which will flow out will obviously depend on the flow rate. If you flow the nutrients through faster, more number of bacteria will flow out. And also, more number of bacteria we have, more chance is there for more number of bacteria to flow out. If you have 10 bacteria versus 1000 bacteria, in case of 1000 bacteria, more number of bacteria will flow out. Simple as that. And why this F by V? You can check the unit. See, F is liter per second. If you divide it by volume and multiply it by M, which has number, okay, right? So M is the number here, actually number per liter density here. So this liter will get cancelled out, and so you will get M, which is number per liter second, and that is dm by dt, right? M is your density, so density by time. How is density changing as a function of time? So the dimensions match up for the first in case of micro. How about the nutrients? So in case of the nutrients as well, you will have the same sort of, since the microbes are the ones utilizing the nutrient in case of this first positive term here, the similar term should appear in terms of the nutrient consumption, right? Because the microbes are consuming the nutrients which are produced. Now, they not might be extremely efficient at the process. Like, for example, if you take a thermodynamic agent, there is some certain efficiency, right? Whatever heat you push into the system, certain portion of that will turn out into effective work. So, in a similar case, if 100% nutrient is flowing in, some fraction of it will be utilized or consumed by the bacteria. So, that is what this alpha conversion factor is doing. That alpha fraction of your total nutrient is being utilized by the microbes which are present. And since it is a consumption process, you have a negative sign. So the growth of bacteria multiplied by the conversion factor with a negative term is the consumption rate of nutrients. And since you're flowing the nutrients, some sort of nutrient concentration is also changing because the nutrients are also flowing out along with the bacteria, right? So you have a similar F by V here. So F by V, so liter per second divided by the volume. Yeah. So F by V. See, in terms of the earlier process which I showed in terms of enzyme and a substrate, it is an enzyme and a substrate interaction which is modeled in terms of Michaelis-Menten kinetics. Here you can think that nutrient is the substrate and bacteria is the enzyme which is consuming that substrate, right? So bacteria nutrient complex. So you cannot call it a complex as such, but since bacteria is acting upon the substrate of nutrients, we can model it in a similar way. So that's why we have taken the substrate. So that was substrate by Km plus substrate concentration. Here it is nutrient by Km plus nutrient concentration in an analogous fashion. Yeah, so we were talking about the nutrient concentration. So nutrients will get consumed in this alpha times the Michaelis Menten. So there is a negative alpha times Michaelis Menten. And since you're flowing in nutrients, so the nutrient concentration in the field, so what you're flowing in, if you consider that as N0, so N0 concentration you're flowing into the system, and N was already present in the system. So what is the amount by which it is increasing? It is N0 minus N times F by V. So N0 minus N will be concentration, and F by V, as you saw, is second, concentration per second. That is dN by dt, nutrient concentration per second. How is it changed? So these two equations we have established. That in terms of microbe density will depend upon the growth rate of bacteria, and it will lower by the flowing out rate of bacteria, and the nutrients will lower by the consumption rate of bacteria and the flowing in rate of nutrients. Now, if I look at this particular system, there are a lot of different units which we have to deal with. See, there are concentration units, there are some rate constant, there are Michaelis Menten constant, volume terms, flow rate terms. So keeping track of all these units might become a bit difficult. So what we can do is we can do a trick called non-dimensionalization. So what basically that does is gets rid of the units. So since these are all variables and parameters and all these sort of things we have observed here, so we can define some new variables. Let us go through this carefully. Let us define time as R into T, which we had defined earlier. Okay. Now, how is this dimensionless? See, R is the maximum reproduction rate of bacteria. So it is some second inverse, the number per second. And your time, which we had defined, was second. So it can be. So this is a dimensionless unit of time. Let us define YT, which is here, as NT by K. So what was the unit of N? It was moles per liter. And K is a Michaelis Menten constant. And what is the unit of Michaelis Menten? It is the substrate concentration at which rate becomes half. So it is the substrate concentration. So it is also mole per liter. So if we divide NT by K, it will also become unitless. Fair enough? So this is your unitless nutrient concentration. We can also do that similarly for a unitless microbe density. So if you do alpha into MT by K, let's look at what that is. Alpha is a conversion factor. So it is anyway unitless. MT, which is their microbe density, and this K which you see here, sort of this particular thing, which is here, has the units of concentration, right? Did I miss something there? No, it's not. Conversion factor. Okay, alpha might not be unitless, but let's take it for the time being that alpha into MT by K is a unitless, sort of unitless measure of microbe density. Okay. And you can also define a new constant which is phi as F by R. So F was your flow rate, which was liter per second. And if you divide it by R into V, so R was second inverse, so 1 by second. And you have V, which is volume. So second cancels out, liter cancels out. Phi is also unitless. It is some sort of a measure of your flow, right? Because you have F by V terms here. And you define Y as Y0, which is the starting concentration of your nutrient in a certain sense, the starting concentration, because you are doing it as N naught by, or rather the flowing in concentration, the dimensionless flow in of your nutrient, because N naught is your flowing in and K is in terms of concentration unit. So you have N naught by K, right? Now, since you have got rid of the units and defined the terms in such a way, you can write these dm by dt and dn by dt in terms of this dimensionless constant or dimensionless quantities. Now, how do we verify? This is xy by 1 plus 1, right? Okay. So let us do, so xy by 1 plus 1, right? So it is alpha m by k into what is y? It is n by k, 1 plus n by k. I'm writing this for dm dt minus f by v, which is, or rather f by rv into alpha by t. This is dx by dt, but dx by dt. And since this t is here, what you have defined t as, so you can take this as dt will be r into t. So 1 by r into del t. And I'll leave this as a homework, or I can show it to you if you have problems facing this in class. If you put in these equations into this dx dt and dy dt, you will actually get to regenerate the dm dt and dm dt. Try it out for once. If you can't do it, I will show you how to do it in terms of the non-dimensional. Okay. Because if I actually I was writing dx by dt. No, I'm trying to get to this from this dx by dt. I'm regenerating t. Hmm. Yeah, it might. Yeah, it really gets a bit. Yeah, you need some bit of it. Okay, you can try it. I'll show it to you. Fine. Huh. Alpha m by k and r will come. You can try it once because I have some more things to discuss. If not, we can do this after the exam, or I will show you in tomorrow's class if you have problem for this topic. For the time being, let us assume that you have converted this into some unitless terms of dx by. So x is your unitless microbe density and y is your unitless nutrient density. Okay. So this is dimensionless microbe density. This is your dimensionless nutrient concentration. Okay. Now, the question which we want to ask is, what is the long-term behavior of such a dynamical system? That if you allow this flow and growth of bacteria and flowing out of nutrients, at very long time, how will this system look like? So at very long time, what you will see as the system will reach a steady state, right? Because there is a certain inflow rate, there is a certain outflow rate, there is certain growth of bacteria, there is flowing out of bacteria. So at very long time, the system will reach a steady state. And how do we define steady state? We said dx dt equal to 0 and dy dt equal to 0. Let me define something here which might be handy a bit later. So whatever is the d dt derivative term, you can write it as a dot like this. Okay. So instead of writing dx dt, we'll write x dot and dy dt will write y dot. Okay. Now at steady state, if we state dx dt equal to 0, what do you get? It is xy by 1 plus y minus pi x equal to 0. From here, if you take out x common, you'll get y by 1 plus y. Right? So from here, you see two conditions. Either you have x, and I should mention this is at steady state. Either this is 0 or this is. And if you rearrange this a bit, you will get YSS equal to y by y. You can do it, it's pretty simple, I think. Sure. If you take phi that side, it will be YSS equal to phi plus phi YSS. You take YSS and then phi by 1. Okay. So by setting only x dot, which is dx by dt equal to 0, you see there are two conditions which arise. Either the steady state concentration of x is 0 or the steady state concentration of y is phi by 1 minus phi. This is from 1. Now if we set y dot equal to 0, what you will see is y by 1 plus y, right? Now this looks a little bit different from the first one, but see, if you have achieved steady state, both dx by dt and dy by dt should be 0, right? So what we can do is, since from the first case we had found out that steady state concentration of x is 0, we can put x steady state equal to 0 in this one as well, because it's a steady state situation, right? So if you put x equal to 0 here, what does y steady state become? Right? And if you put, so this comes if you put xss equal to 0. So you have generated one steady state combination, which is x is equal to 0, y is equal to y0. This is one steady state, right? Because you put both dx dt and dy dt 0, you found out what xss is, steady state of x, and at that value of steady state of x, what is the corresponding value of y steady state. And see, if you have the 0 value of xs, you will get y naught as the y. And this makes sense, right? If there is no sort of microbe density at year time, you will get the whatever you're flowing in that. And if you put this phi by 1 minus phi in here, so minus x to phi by 1 minus phi by 1 plus phi by 1 minus phi plus phi into and sitting some of the steps here. But if you solve this, what you will see is this comes out to be you can try this. It's not very difficult. So this is your second combination for steady state. That when excess is y naught minus phi by 1 minus phi, this is okay. So there are two possible steady states in this: 0 and phi by 1 minus phi, or y0 minus phi by 1 minus phi and phi by 1. So okay, let me just note it down here. Hmm. 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 What we have defined phi as F by R. So it's a flow rate, it's a reproduction rate, and it's a volume. All of them are positive quantity. System. All are positive quantities. And one thing which I must mention before proceeding here is the way we have defined this condition is phi will lie between 0 and okay. So now let's do something. Let's plot this plot this. So in this direction I have x, and in this direction I have y. Okay. Now let us see what we do. By setting this to 0, we got the first steady state. If you look at this carefully, this is some function of x and y, right? And when you set this to 0, you got this particular equation. This is some other function x and y. Now what I can say is, since this is a function of x and y which we have set to 0, we can represent this in terms of a line. So xy by 1 plus y minus I is equal to 0. So if I show you, I have the picture. If once you do that, if you can see, so xy by 1 plus y. So if you look at this violet lines, xy by 1 plus I minus phi x equal to 0. You saw there is 2%, right? One is x equal to 0. So this is, as you can see, so the y-axis, which is here, is the x equal to 0 line. So that is one solution. And another one is y equal to phi by 1 minus, which is the horizontal line. And focus on the first coordinate, you don't need to worry about negatives. So by setting this x dot equal to 0, you get the violet lines. One is x equal to 0 and one is y equal to phi by 1 minus 1. Since phi is constant, you will get a straight line. If you set the second term, which is dy dt equal to 0, so that is the first red line which I have shown, that minus x over 1 plus 1 plus phi, y not phi. And I have assumed some arbitrary values of phi and y naught. You'll get this particular line, the red line which you see is here. So the red line which you see follows, and it has cut this violet lines at 2 points, right? So think carefully. One line, so if I show this now, let me draw this here. So this is one line. This has come from x dot equal to 0. This has come from y dot, right? Now, and also this particular line which has come as y dot equal to 0. Now you might think that why two lines from y dot equal to 0 and one from x dot? If you look at that picture, that x dot, the other violet line, was completely in the negative domain. And you cannot look at like negative nutrient concentration. So we are omitting that. There are two lines mathematically, but we are looking at the positive. Now thinking, why have I defined this particular point as steady state 1 and this as steady state 2? Why this intersection? Look at this carefully. One line came from dx dt equal to 0. Another line came from dy dt equal to 0. So when both of them are simultaneously 0, that is your steady state. Okay? So I have defined this as steady state 1, which is this one. Steady state. Okay. Now let's look at something interesting. So at these, along this particular line, these are 0, right? So on either side of these two lines, it will be positive or negative. So suppose this is x dot equal to 0 line. On one side of this, x dot will be positive. On the other side, it will be negative. Similar for y dot. On one side, it will be positive. On one side, it will be negative. Since this is not a quantitative one, I'll go back to this particular picture. So, for example, this red line, which you see here, is the y dot equal to 0 line, right? So, let us take one particular point, which is 0, 0. Okay. Now, see, if you put 0, so the y dot equal to line is the red line. Right? If you put 0, 0 in that equation, what is the value of, so don't put that equal to 0, but that is dy dt, right? Dy dt is the red line, which I have plotted, dy dt equal to 0. So if you put x equal to 0 and y equal to 0 in that equation, what is the value of dx? 5 times y naught. See, phi is positive as I mentioned, y naught is positive. So in the red line, if this particular origin point, it lies on this side of the red line. So if the value of y dot is positive here, that means the entire domain which is on the origin side of the red line, in all those cases, y dot is positive. As along the red line, it is 0. And once you cross over to the other side of the red line, y dot will be negative. Okay? Yeah. So what you're doing, the first equation is dy dt, right? You put the value of origin, let's say 0, 0 in the first equation. So we will remain with dy dt equal to 5 times y naught. Phi is positive, y naught is positive. So dy dt is positive at the origin. And since I mentioned that it will switch signs across the line, so that is how it happens. So I can say that towards the origin of the red line, wherever you see, y dot is positive, and beyond, like, away from the origin of the red line, y dot is negative. And for x dot, what you will see, okay. So below the line, so in this case, okay, let me put it in color. It was red and this was violet, right? So let me draw this in. Why don't I? Okay, let's move this screen. So y dot, whatever is towards the origin is positive from the line, and beyond that line, it is negative. If we look at x dot also, in this particular case, so the violet line, let's look at the horizontal line itself because it will be easier to denote in that. To do something, put the point, what do you want to put? No, no. You can put 2 comma 2, 2 comma 2. Put this in this equation. This is dx dt equation. You put 2 comma 2. Put it in this equation. And put 5 years points, right? Because that's how the graph has been drawn. So by 3 minus 4 by 3 minus 4 minus this is going to be 4 by 3 minus 1 positive. So so above the green line, x dot is positive. Below the green line, x dot is negative. Now, Let's take a look at how it will look in the XY domain. If you take a point here, y dot is negative, x dot is positive. So how will the system move in the XY space? The y amount of y component will decrease and x component will increase. So it will move somewhat like this. Right? But now it has faced a critical point. Beyond this line, x dot is now negative and y dot is still negative. So now it will start coming like this. Now it has faced the y dot equal to 0 line. But x dot is still negative. But now y dot is positive. So x will decrease, but y will start to increase. So it will move like this. Now it has faced this domain. Now, what will it see? Here, so in this particular domain, which I'm mentioning, might be because of the highlighted display. So in that domain, y dot is positive and x dot is also positive. So it will start moving in this direction. It is a bit slow, but you will continuously it will move like in this direction. Now again it has crossed. It will move like this, and again like this, like this. So you can see the XY components are slowly collapsing onto this point. So no matter wherever you start with, so I had taken this particular point, whether you start here, whether you start here, whether you start here, if you draw this sort of lines, which are called vector fields, and this XY is called the phase portrait. So names are not important, the significance is important. So in the XY plane, if you draw the arrow that in the direction of the movement, you see no matter wherever you start, you are collapsing onto this point after infinite amount of time. So this SS2 is called a stable steady state. No matter wherever you have started in this x and y amounts, after infinite amount of time, the system will collapse onto these values of x steady state and y steady state. So that's why it's called the stable steady state, because it is drawing, attracting everything, no matter where you start, it is attracting it to this final point after infinite. And if you look at this SS1, which we are denoting now, see, for example, hypothetically, if you have started exactly at this point, then there will be no movement, right? Because at that value, x dot and y dot both is. If you take a point which is slightly away from this, just like infinitely away, it will start moving away, and ultimately everything will fall back to this point. So although this is a steady state, this is called a unstable steady state. Because it is a steady state, but if you slightly move the system away from that point, it will completely move away and never return it back to it. So there are two states in the system. One is a stable steady state and one is a unstable steady state. Understood what is why is it stable and why is it unstable? Now if you look at it carefully, the way I have drawn this is I have made an assumption that this was the phi by 1 minus phi, this horizontal line, this value. And this value had been y. So if you look at this particular graph, see I put y not equal to 2.5. So you see that 2.5 and this phi equal to zero point 0.5. So phi by 1 minus phi is 0.5 by 1 minus 1, right? So I've assumed that the starting value of that y dot equal to 0 line, y naught, the y intercept of that is higher than phi by 1 minus phi. The way I have drawn in this particular graph. Okay. So in this one, y naught is greater than phi by 1 minus phi. Okay. What will happen if y naught is less than 1 phi by 1? If you draw a similar picture again, you will need some negative axis here and some negative axis here. Zero positive x, zero positive y. If you can't draw it by hand, it's fine. We'll take the help of graphing software again. So phi by 1 minus phi is 1, right? Because I have kept phi as 0.5, 0.5 by 1 minus 0.5. What I'll do is, if I drop the y naught below 1, that will be y naught is less than phi by 1 minus. So I'll just drop this y naught value to less than. And if you focus only on the positive part, what you see here is that this point of cutting, so this particular point, so the point where it is cutting, it is beyond here, right? Right? It is still cutting at 2 points. One point it is cutting at x is equal to 0 and y is equal to that phi by 1. Right? And the earlier one, which was steady state 2, when I had kept y naught higher, now it has moved to the negative 2, which is unattainable by the system. So the only possible steady state which can be achieved by the system, practically speaking, is the one here. And here, so this 0, 0.4 for our value. And I'll leave the exercise up to you because I've shown one here. So the line looks so much like this. It's 5 and the x dot this. This will be 1, which is unattainable, and this will be your x is equal to 0. You can actually see that now, if you draw the vector fields, they will all start converging on this particular point. No matter where you start, they will show the similar behavior and start collapsing on this. You can draw that graph in any graphing software and just put the values like we did. You put any 2, 2, you put origin and check the value, what is dx dt, what is dy dt, and move your pen accordingly. And you will see that now, earlier what was unstable, now it has become stable. And what was earlier stable, now has become unstable, or unattainable, unstable, whatever you want to call it. If you consider mathematically it is attainable, you will see that the lines will move away from that point. You can try it. Mathematically, you can try it. So, we have observed a very interesting thing. When y naught is greater than phi by 1 minus phi, your steady state 1 is unstable and steady state 2 is stable. But when y naught is less than phi by 1 minus phi, your steady state 1 is stable, steady state 2 is unstable. And for reference, steady state 1 is that 0, y naught, steady state 2 is y naught is phi by is phi. One minute, right? So I can do something like I want to see what is the value of my steady state microbe density as a function of y. Okay. So phi by 1 minus phi is sort of the turnover point, right? So when y naught is less than, okay, let me put one line here, which will be, let us assume this is the phi by 1 minus phi. And it is in the along the y coordinate, so it's y naught, right? That's why it's a vertical. Now, when y naught is less than this value, what is your steady state? Or rather, what is the value of your steady state concentration of X density? Zero, right? Because that is the steady state. So up to this point, this is your steady state. Once you cross over to when y naught becomes greater than phi by 1 minus phi, what is your new excess value? Here. And since we are plotting this as y naught, so this is y equal to mx plus c equation. And since it's it's y naught minus phi by 1 minus phi, and phi is between 0 and 1, so this is positive quantity. And so the intercept will be negative. So I'm leaving out the intercept for now. The line will look something like this. So before phi by 1 minus phi, one state is stable, one is unstable. As soon as you cross over to higher value, which is greater than phi by 1 minus phi, the system switch their stability. So what was earlier stable now has become unstable, and what was earlier unstable now has become stable. So at this particular point, there is an exchange of stability, right? And this picture which I have drawn here is called a transcritical bifurcation. Why a bifurcation? Because there are two states in the system. Earlier one was stable, now the another one is stable. So at that particular point, it is bifurcating into two regions. Yeah, was anyone saying something? So the system exchanges stability between two steady states. This is called a transcritical bifurcation. And this is the beginning of something which we'll deal with called bifurcation analysis. This is one type of bifurcation. There are many different types of bifurcation which arise from different interactions. The way we write these equations, how they're interacting with each other, will give to different types of bifurcations, which we'll look into the coming classes. This is one of the most basic types of bifurcation, which is called a transcritical bifurcation, where there are two steady states possible and they exchange their stability at one particular point. So if I ask you to draw the vector field at this particular point, so it will look something like this: that wherever you start from, the system will start collapsing here, right? And once you cross over this point, it will start collapsing from here. And it will actually, if you do so in this portion, the first steady state has now become unstable. So actually, it will start moving away. I can show you in another color dotted line how this system. So if I draw the unstable portions, so usually the unstable portions are denoted by dotted lines. Right. Right. You can see that as we start. Okay. So you can see what was red up to here, it is stable. Beyond that, it is unstable. And the red, if you look from the right to left, so it was stable. And beyond this, it is unstable. And the arrows correspond in the phase space. Clear? Okay, we'll keep the instructions up to here right now. Let's take a short break, and I have two small problems which will take 15-20 minutes. We can discuss this. Okay, you can all take a break, and after 10 minutes, you can join back. We'll join back after 5 minutes. Take a break, and we'll solve two small problems, which will give you a better flavor of the bifurcation. Okay, so let's get back and solve some interesting problems. So since we had talked about these bifurcation, these vector fields, space portraits, steady states, let us do some simple problems and try to see in a physical sense how these bifurcations exist around us. Okay, let's do this particular problem. So Wedelst in 1838 proposed a population model, which is called the logistic growth model. And anyone who has studied NCERT class 12 biology book must have read the Worrell's fault logistic growth model. So the equation is something like this, which is dA and dt. So n is your population. It depends, or rather grows. So r, there is some certain growth rate r, n into 1 minus n by k. So it's And this is a realistic growth model because if you see populations around you, no population grows infinitely to a arbitrary extent. There has to be some capping because there is strain for resources, there is physical constant for any population, so it has to survive as well. So it cannot grow uncontrollably. So there is an n term which is a linear increase term, and to counter that, to keep the population under check, you have this 1 minus n by k term, and k is called the carrying capacity, which is the maximum amount of population that can be sustained by one particular system. So that's evident also. If you put n equal to k, you say d and dt will go to zero. The population will stop growing. So that becomes one of the first ones. So first is you have to find out the fixed points of the population growth model. Now, what I called as steady states in terms of this nonlinear dynamics and mathematics, it is called fixed points. It is essentially the same. So to find out the fixed points, simply we have to put this d and dt equal to 0. So two situations are possible. One is n equal to 0, which is the trivial solution. If there is no population, then who will grow? Another one is 1 minus n by k equal to 0, which gives you, it is not exactly a dimensionless. You can consider it's a population, right? So it's a number, and r is a growth rate. So you can consider like d and dt will be some number of people per unit time. And see, one n by k, so k is a capacity and n is also population, so that gets cancelled out. So r into n. R is growth rate, so some time inverse, n is number, so population of it. Not necessarily will show. It's non, no, it can be anything. Will show. That's part two of the. The first one is fixed point solution. So there are two steady states. One is n equal to 0 and one is n equal to k. Now 0 is a trivial solution, as I mentioned, and the more important solution is n equal to k. So at infinite times, the maximum population, or rather the only population value which can be reached in a certain sense is the carrying density. So this is the first part. There are two fixed points, n equal to 0 and n equal to 0. The second part is interesting mathematically. You have to show that the population will always approach the carrying capacity if you start off with a finite initial population. So N0 is the initial population. Now we can do this in two ways. First, let's do the brute force way, which is integration. So you have dn dt equal to r in 1 minus. If you do some rearrangement, you can write dn dt equal to I've just rearranged the terms and accounted for the negatives. So now this is a partial fractions problem, right? And how can we split it? We can write it as but if we do this, there is an extra positive k in the numerator, right? So we'll do 1 by. So if you proceed in the next step, this k gets cancelled out from both sides. And if you divide, so it becomes now you put integration. Your time will be 0 to some time t because you're trying to look at population as function. So when your time is 0, your n is n naught, and the time t will be some value of n, right? And so if you do this integration, you would do it is growth rate is or this r, which is what is mentioned as growth rate here, it is fixed. Here up to this. So if you expand this, what you will get? N minus k by n into I'll just skip one step here. If you put, so it is ln, once you put n minus ln n naught. So now we can remove the log by taking e to the power n. Yes. So n minus k by n into proceed further. This will give you, I can divide this, taking it here, and I'm taking this one here. So I can write it on this. N becomes 1 by 1 minus 1 minus k by n naught e to the power minus now if we rearrange the terms a little bit, what we will see here, this will become Let's skip one step in between. Did everyone fix this? So now you see, if you have some positive n naught, and if you put e tends to infinity in this equation, what is your value at n when t tends to infinity? So this exponential term will become 0. N naught cancels out. No matter whatever your starting population, other than 0, after infinite amount of time, the system or the population will stabilize at its carrying capacity. There is only one possibility. Clear? Now, if you don't want to do this integration, you don't like calculus, there is another method to do this. What I'm going to do is draw this graph. How should this graph look like? When you multiply this, it will be Rn minus Rn square by k. So it is some sort of y equal to x minus x square. So there is a quadratic term involved for sure. So it will have some sort of a parabola like shape. And since you know that d and dt will be 0 at n equal to 0 and at n equal to k, that is where the parabola will cut the x-axis. So parabola will look something like this. Right? Now, if I want to do something like this, if I am somewhere here, what is the value of d and d? Positive. So n should increase. So the system will move here towards infinity. If I am somewhere here, what is the value of d and d? Negative. So n should decrease. So you see, just like we had that steady state, which was a stable steady state, this n equal to k is a stable steady state for this population. And that makes it very clear that since this is positive and here it is negative, this is unstable x equal to 0 or n equal to 0. So here prove the first and second statement just from a graph. Wherever the system will cut to 0, that is your fixed point. So there are two fixed points at 0 and k. And from the diagram also you'll see that no matter where you start, apart from 0, the system is always reaching towards. Yes. So if suppose you're here, if you move just slightly towards the right, what is the value of dNdt? Positive. Okay. If you're here, somewhere here, and if you move slightly towards the right, what's dN? Positive. So n will increase. So increase. So it moves away from that towards the right. And if you are slightly towards the left of that, n will decrease. So it will start moving. So it's the unstable state. If you try to reach it, it will repel you away from that point and push you back to the stable state. It's, and some diagrammatic notation. So whatever is a stable steady state, if you want to mark it in a diagram, you do this by complete dot. Whatever is unstable, you leave it as a empty dot. That is normal convention. How it is. So if you want to draw, this will be something like this, empty. This will be shape. So that tells you that this is unstable, this is stable. So we have modeled an entire population just based on one single equation with two multiplicative terms. Now this was the model and the steady state analysis. Now we can look at the bifurcation diagram of such a model as well, which has a similar functional dependence. What do? Let us say a population growth model where x is the population is dx by dt, and it is x square minus some rx. Now, how does the graph look? If you don't want to do it by hand, let's go to Desmos. Let's take a new blank graph. X square is x. So you see, if I keep changing r, when r is negative, the graph looks something like this. At r equal to 0, it's like this. And when I increase r, it becomes like this. Right? So there are three cases which happens. When r is less than 0, the situation was like this. When r is equal to 0, the situation is like this. And when we have r greater than 0, the situation is these are all dt. You can we saw that. Right? One of the point will always pass through the. How do we know where it will pass? So we can see where it will be 0. So either it is x equal to 0 or x equal to r. So when r is less than 0, let us take this particular case. Can you tell me from the graph? So this is on the left, the point where it is cutting, it is x equal to r, right? And that is x equal to 0. So there are two steady states in the system, x equal to r, x equal to 0. The point or the steady state at x equal to r, is it stable or unstable? Think carefully. Yeah, for the left, for this point. So I am marking this point whether it is stable or unstable. It's a stable state, right? Because see, if you are slightly towards the left of that, the density is positive, so x will increase. And if you're slightly towards the right, the density is it's a stable state. So obviously x equal to 0 will be an unstable state. Because you see, towards the left of it, it is negative, so it moves away from there. And towards the right. So do you see a simple pattern emerging? Wherever dx dt or dn dt or any dot cuts your axis in a negative direction is a stable state. Whenever the line will cut your x-axis from top to bottom, it will be a stable state. And whenever it will cut from bottom to top, it will be an unstable state. Right? Now, the interesting case comes at r equal to 0. So for r equal to 0, if you're slightly towards the left, the density is positive, so the arrows will start coming here. If you're slightly towards the right, then also it's positive. So it also moves here. So this is a special type of point which is called a half stable point. It is neither completely stable nor completely unstable. It is called half stable point. Usually you won't get to see this half stable point in most of the discussions we'll do, but this is one such interaction which actually, one such case which actually. And for r greater than 0, from the trick I showed you can see that this will be stable. And definitely this will be unstable. Half stable. Hmm. So again, you see, this is exactly like the transcritical bifurcation we had seen earlier. Depending on one parameter, the two steady states are exchanging the stability. So in this case, if you want to draw the bifurcation diagram, where R is a parameter, and we are plotting X steady state here. When R is negative, or when R is positive, 0 is your steady state. When R is negative, so this is also transcritical. The system has exchanged its stability at this station. Okay. Okay, you can keep it to here. Okay. Yeah, so let's keep our discussion till today to this point after transcritical bifurcation. If you want to learn more about uh, if you want to learn more about these bifurcations and sort of thing, the best book is by Steven Strogatz called Nonlinear Dynamics and Chaos. We'll mostly be following that particular book and we'll solve more examples in the next class. So see you tomorrow at the same time, 2 p.m.